applications as opposed to using the inhibitors?
Yeah, that's a great question. So right now we utilize CRISPR-Cas9, which is a technique that really makes it a lot easier to do gene editing in a lot of the research that we do. As a particular therapy, most drug companies, and what I was mentioning before, where I think we have the best shot at trying to get a therapy, is not to do the gene therapy, but is to do the kinase inhibitor therapeutic approach. And I mentioned before, it's because there's so much expertise in doing that. There's past experience within the oncology field that this is an approach that could work. And we think that's our best shot. But we're definitely keeping an eye on CRISPR-Cas9, and we do use it quite a bit for our research.
We do have a question from social media, so we'll go start here.
Hi, yep, I'm Emily from NASA Social Media. This question comes from Twitter. Are the proteins tested extracted from a particular cell or product?
Yeah, the proteins are human proteins. That means the sequence is the same as the one for human proteins, but we make those proteins in insect cells with the baculovirus system. And that's how we can make a lot of the protein. Extracting it from human source will not be sufficient.
Thank you. Do we have any other questions in the room? We have one question toward the back. Please wait for the microphone to reach you.
I'm just curious, with the results that you guys are going to end up with, hopefully some positive stuff, are there other disease organizations that are going to benefit from this, that are kind of watching you guys to see what your results are?
So in case we will get the structure, we will of course publish it as soon as possible and make it available for the public.
Yeah, definitely everyone in the Parkinson's field looks at other neurodegeneration fields. We monitor what's going on in Alzheimer's. I think they monitor what we're doing also. At the highest level, that's the type of monitoring that we do to see where the similarities are and where the differences is. Specifically with this experiment, I'm not sure how this will be affecting other disease indications, but as Sebastian said, we try to get the information out and people can freely access and see what we found.
Thank you both for your time. Thank you. Our next highlight is the effect of microgravity on stem cell recellularization, also known as the lung tissue experiment. For that, we have a video ready for you.
We've created a model of the human lung that has most of the cells in it that a lung would have, and we're going to use this model to evaluate what happens to your lung on long-term spaceflight. We're using stem and progenitor cells. They're the cells that give rise to the cells that allow you to have gas exchange and breathe. The larger mission is to eventually someday be able to, instead of make pieces this big, is to make a lung this big for transplantation for somebody. If you're going to look at what atoms do, if it affects a molecule, that alters a cell, that changes how a tissue responds, that alters how an organ functions, that changes how you function, you have to be able to walk it back and say, I can look at it at these small levels and build this picture of what's happening in a person. That all may be very different in space because microgravity influences everything that a cell does. And the opportunity to have a chance to send an experiment to space and get data back is huge for a scientist. It happens once in your lifetime.
Unfortunately, Dr. Nichols was unable to be with us today, but we do have Ken Shields, we'll bring him back up from CASES, who to answer a few questions about the lung tissue investigation.
Questions are not required.
Do we have any questions in the room? Do we have any questions on social media? Once again, you can submit